Welcome back. Welcome back after we have understood that the mechanisms behind CCD5 and AEX3 are vaguely understood as involving Nectin and UNC5 somehow and involving axon transport somehow. I return with the most exciting question. Okay, but why? How? What? This was the question that I was most excited about when I came to this lab. How does CCD5 affect the ventral nerve cord? These ventral nerve cords look very weird. Why? Why do they look weird? And in what ways? What paths did these growth cones take? And how were they guided? So, the first step to understand this is by fully describing how weird the ventral nerve cord actually is. And here we have some beautiful pictures communicating that. Aren't they so pretty? We see all of the range of defects that ABG, our pioneer axon, can have. It can cross the midline. It can start on the wrong side and correct itself. It can start completely randomly in the wrong place, come to the ventral nerve cord, and extend in weird ways. It can, again, start in completely the wrong place and extend in weird ways. And it can just wander off confused and branch and then come back. It usually does come back, though, which is nice. When we're talking about command interneurons, we see another enthusiastically wide variety of defects. There's the ones that I want to highlight are some cross-left, where a subcategory of the command interneuron axons will cross the midline and then cross back. And all cross-left, where all of them will cross the midline and then come back. Or maybe some of them come back in this case. And of course, both of these things happen in motor neurons as well, where all can cross or some can cross. So, meet a table. We have entered results time. Here we see the total axon defects in a wild type, a CCD5 single mutant. Remember, all of the stuff we've been talking about was in that nitogen 1 null background. Here's our nitogen 1 single mutant, and here we see our double mutant, which is the one that we care the most about. And interestingly, there's a pretty intense baseline level of random going on in motor neurons in wild type. And in CCD5 single mutants, we also see an elevated level of motor neurons, significantly more than wild type. So CCD5 is doing some stuff on its own in the context of motor neurons, even without the nitogen 1 null. So when we see nitogen 1, we see this lovely baseline 20 to 30% defects in everything. And our double mutant, we see double everything. There's this huge amplification going on. So in conclusion, ABG and follower axons are legit misguided. But there's another interesting thing going on here. What's the big difference that you see between the purple and the red? Give it a moment. The red looks like AVG. Command interneurons look like AVG. And the motor neurons don't. Motor neuron axon defects seem to be affected by that CCD5 single mutant defect in a way that command interneurons aren't. So that's already an interesting thing. My next gut question here is, are command interneurons responding to CCD5 at all, or are they just following AVG? What is the relationship here? Are motor neurons following AVG at all, or is this just an additive effect where 30% are messed up in nitogen 1 defect, nitrogen 1 null, 30% are messed up in CCD5 null. Together, they have almost twice as much, right? Maybe there's no following going on at all. So to test that, nah. I spent a lot of time drawing pictures of worms. So in these worms, there's a fluorescent marker for ABG and a fluorescent marker for command interneurons. And in these worms, there's a fluorescent marker for ABG and a fluorescent marker for motor neurons. And I sat down and drew hundreds of these with pencil on paper to try to figure out the relationships between these defects. And I ended up sorting them in this very specific way. So I sorted them as either crossing a follower axon, crossing the midline at the same point that ABG is crossing the midline, or not, and a follower axon crossing fasciculated. So all of the axons in a single fascicle crossing together. The fact that all or some variable. And so you can imagine this as a grid with fasciculated versus defasciculated and with ABG versus independent as creating 4 different options. And these are those 4 options. And in here we see the gray is the nitrogen 1 mutants, which are going to be our control, that single mutant. And then the color is the double mutant, CCD5 nitrogen 1. And when you look at the command interneurons, you see that the only significant value here is a fully fasciculated group of command interneurons crossing the midline with ABG. None of the other ones are significantly different from the nitrogen 1 control. So you see that? There's everyone crossing. Every last red one is crossing the midline, the same place that the green one is crossing. Now there's different stuff going on with the motor neurons. When you look at fasciculated, again, you only see changes with ABG. So when you're talking about all of the motor neurons coming together, like this, crossing with ABG, that's significant. But when it's fasciculated, there's no significant difference when it whether or not it crosses without ABG. However, when we're talking about this sort of thing, where the motor neurons are leaving other motor neurons to cross the midline, then they don't care. There's just more going on. So, do the follower axons rely on the pioneer? Well, fasciculated follower axons do, definitely. If the follower axons are all fasciculated to each other, they're also going to stick to the ABG. And if the ABG crosses, they're going to cross too. However, the motor neurons cross on their own, with and without ABG. And that's different. That's not true about command interneurons. And that brings us back to what we saw earlier with the motor neurons being affected directly by CCD5 when there was perfectly functional nitrogen 1. So what does it all mean? Yeah, brings us back to that. Look, it's a whole story. Oh, my research is the whole story. We see CCD5 is needed for correct ABG axon guidance. It's also needed for correct motor neuron guidance directly. Motor neurons are depending on CCD5, independent of ABG, for their guidance. That's shown here. We're also seeing that that is not true for command interneurons, that the defects that we see in command interneuron guidance result directly from command interneurons following ABG. And it's not at all affected independently of ABG. We're just seeing defects as a result of AVG midline crossing. We also see this lovely fasciculation observation, that motor neurons and command interneurons are more likely to follow AVG across the midline when they're already fasciculated with each other. And to me, that says something about the act of fasciculation, that if the follower axon is fasciculated to its other follower axons, it's also going to be fasciculated to its pioneer. That fasciculation is the same regardless of whether it's a follower or a pioneer. And having that fasciculation intact makes it more likely to stay with the follower, or with the pioneer. So let's talk about motor function. The motor system does all of its processing in the ventral nerve cord. And the ventral nerve cord in these double mutants is really messed up. So, do CCD5 mutants have motor defects? It makes sense that they would. If all of these synapses are supposed to happen en passant, and a fair number of the motor neuron axons are wandering off without staying fasciculated, that might have a functional repercussion, right? To test that, we used the multi-worm tracker, courtesy of the Rankin lab at UBC. The multi-worm tracker records many worms all at once, at rest and in response to tap. So we assessed free movement, where if you leave worms alone, they will spend a portion of their time still and a portion of their time moving forward. We also assessed avoidance response. When you poke a worm or you poke a plate with a little thumpy thing, they reverse. And then they move forward at a new angle as if to avoid an obstacle, which isn't shown in this GIF because there are limited GIFs in the world. But it is a thing that happens. There's a reversal and then there's a forward. And we can measure those by recording them on a camera. We did that. Here are some results. And for context, the red is a negative control. These are worms where the AVG fails to differentiate. So at the worst, least healthy AVG response situation, we should see that phenotype in Lin11. And we do consistently see Lin11 null worms as the worst performing of our worms. So that kind of gives us a floor of how bad it can get, if that makes sense. So I want you to spend some time looking at these graphs and try to figure out what they mean. What are the effects of these mutations functionally? This is that free moving plate, and this is just how much time they spend moving forward. So wild type spends 30% of its time moving forward. Lin11 spends 10% of its time moving forward. And significant difference happens when these error bars do not overlap. So Lin11 is spending significantly less time moving forward. So with that in mind, ask yourself, do we see any defects in CCD5 single mutants or CCD5 NID1 mutants moving forward, or their proportions moving forward? Do we see any defects with their likelihood to respond to tap? Do we see any defects with the amount of time they spend reversing? And I want you to look at these while thinking about this. Okay. So let's talk about it. What does it all mean? So this is significantly decreased. Our single mutant CCD5 is significantly less likely to respond to TAP than wild type or NID1 single mutant. It's not significantly less likely to respond to TAP than our double mutant, but our double mutant is also not significantly different from our wild type, because statistics. When we look at reversal duration, we see that our double mutant is significantly less, spends significantly less time reversing in response to TAP than our wild type. But this does not reach significance, just barely, but the double mutant is not spending significantly less time than the single mutants, either of them. So what does it all mean? Is the double mutant functionally affected? Is the double mutant not functionally affected? Do we really not have enough information? What do you think? So my original one that I wanted to put for C, or maybe D, is none of this matters, statistics is a lie. So I picked D. We've done too many statistical analyses, and now we're randomly throwing things at the wall to see what sticks. And none of it matters, and everything is broken. This is why I don't do behavioral studies. All right, so let's talk big picture. These guidance defects aren't affecting motor function as dramatically as they're affecting the ventral nerve cord midline crossing. That's clear. Ventral midline, this double mutant has over 50% defect rates in the ventral nerve cord. But they're barely affected functionally, looking at these behavioral parameters. Maybe this is because AVG defects don't affect ventral nerve cord fasciculation. Even though the AVG crosses the midline and all of the others are crossing the midline to follow it, that fasciculation is intact, so all of those en passant synapses can still form and the motor system isn't affected. Or maybe other redundancies exist to ensure correct behavior. Or my favorite one, even these tiny defects are dramatic. And being even a tiny bit bad at avoiding danger would be deadly in the wild, which is why there are so many layers of safety preventing defects in the ventral nerve cord and in this system, because that spider needs 8 legs in the wild. In the wild, it needs to be able to be maximally dexterous. It needs to be able to run and hunt and hide and respond to stimulus as effectively and nuanced, in effective and nuanced ways that there's no way for us to measure in the lab, because the lab is such a controlled environment. So we're only picking up a tiny little bit of these behavioral defects that could be incredibly important in the wild. So these are the results of my research. There are more questions than answers, which is the whole point of science. The more you know you don't know, which is why it's fun. Welcome to science, welcome to research, welcome to academia. This is a summary slide. Today, we learned about how to conduct a genetic screen and an enhancement screen, or at least we reminded ourselves that we already know. We learned about redundancy and how it complicates research and ways to get around it. We also talked about how mutations affecting axon transport might affect navigation. And we talked about 2 different ways to assess the effects of navigation errors on the ventral nerve cord function. We talked about looking closely at axon pathfinding, and we talked about behavioral assessment. But mostly, we talked about how to ask testable questions and how to find testable answers that lead to more questions that you can test. here have some vocabulary words. Everything is clearer with vocabulary words.